Hello, my name is Derek Reiman, and I'm a PhD candidate at the University of Illinois at Chicago Department of Bioengineering under Professor Yang Dai. Today I will be talking about MiMeNet, a framework we have built that is used for exploring the microbiome-metabolome relationships using neural networks. A microbiome consists of microscopic organisms cohabitating in a shared environment. Of particular importance is the microbiome of the human gut. Many studies over the last decade have linked various metabolic diseases to the human gut microbiome. Even more recent work has begun to identify the importance of bacterial metabolites and their impact on host health. Microbes have been shown to be able to synthesize their own metabolites, utilize and modify host metabolites, and there are even cases of host microbe co-metabolites. However, the exact nature of microbe-metabolite interactions within a host are still not well understood. And by being able to identify microbe-metabolite interactions, especially in terms of the development of disease, we can understand better how the microbiome affects the health of the host, and, in addition, the knowledge can potentially provide insight in the development of therapeutic treatments. Previous work has modeled microbiome-metabolite interactions using a priori knowledge of mechanisms and stoichiometry. Here I describe two major approaches: predicted relative metabolic turnover, or PRMT, and flux balance analysis. PRMT is a computational analysis tool that uses the changing abundance of functional genes or enzymes in metagenomic data to predict the capacity of that community to consume or generate metabolites. For example, here we can see a vector of microbe abundances on the right, and it is being multiplied on the left by a matrix that defines the abundance of enzymes, resulting in a vector of enzyme abundance values. This is then further multiplied on the right by a matrix that describes each enzyme's effect on the metabolites, giving a final vector of metabolic turnover. Flux balance analysis, on the other hand, formalizes a system of equations describing the concentration changes in a metabolic network as the dot product of stoichiometric matrix and a vector of the unsolved flux values. Although they have been successful, these methods share a common drawback of relying on accurate network and reaction annotations, and this makes it challenging to uncover new interaction mechanisms. More recently, a data-driven approach was designed by Malik et al. Called MELANPAN, which uses linear elastic net regression to predict a metabolite's abundance for microbiome data. As a data-driven approach, it no longer requires the a priori knowledge of annotations, allowing it to learn new relationships. However, the model only considers linear relationships and can only model one metabolite at a time, reducing its scalability. To address all these challenges, we set out with the objective to construct an integrative microbiome-metabolome data analysis framework that can predict metabolites for microbial features using nonlinear relationships, model the entire metabolomic community at once, and derive microbe-metabolite interactions in a data-driven manner. We call our framework MiMeNet. MiMeNet takes in paired microbiome-metabolome data as its input. We then do some pre-processing and filtering, and then transform the data into compositional data. Next, a neural network model identifies metabolites that can be well predicted for microbial features. And by using a neural network, we can learn nonlinear relationships as well as model the entire metabolomic community at once. The weights of the trained neural network are then used to construct micrometabolite interaction scores. The interaction score matrix can then be biclustered to identify microbial modules and metabolomic modules, and we can use these to construct a module-based interaction network. For this presentation, we will look at how MiMeNet works on some inflammatory bowel disease data. We used paired data from the Prospective Registry and IBD study at Massachusetts General Hospital, which we will call PRISM. The data contains 123 patients with IBD, 34 healthy patients, with a total of 201 microbial features at species level, and 8,848 metabolites, of which 466 were annotated. In order to identify well-predicted metabolites, we perform 10 iterations of tenfold cross-validation. We predict the metabolomic profile from the microbial features. We then evaluate each metabolite using the Spearman correlation coefficient. To determine the correlation cutoff, we perform a similar experiment of 100 iterations of tenfold cross-validation, except this time we shuffle the samples in each dataset separately for each iteration. This gives us a background distribution of correlation values, and we can set a cutoff at the 95th percentile. So here on the left, we can see what the two distributions look like. Our observed correlations are shown in orange, and the background is shown in blue. The red line shows the 95th percentile cutoff, so any metabolite to the right we consider well predicted. Next, in the middle, we show how MiMeNet's prediction compares to MELANPAN, and we notice an overall improvement. We believe this is from the neural network's ability to model nonlinear relationships, as well as the benefit of multitask learning through predicting the entire metabolomic community. Note that we only compared the 466 annotated metabolites, but MiMeNet was trained on all 8,848. Lastly, on the right, we are showing the top correlated metabolites. We find that even though MiMeNet was given no information about IBD status, many of these metabolites have been associated to IBD in previous studies. Now that we've trained our models and showed their predictive power, we want to interpret what is going on within the model. For this, we use Olden's method of variable importance for neural networks, and it works quite simply by multiplying all the weight matrices together. This results in a single matrix that, for us, will have each row representing a microbial input and each column representing a metabolite output. The higher the value of a position indicates that the microbe has a positive influence on the generation of the metabolite, and likewise, a negative value means that the microbe reduces the value of that metabolite. We generate a single interaction matrix as the mean value of the matrices from the 10 iterations of cross-validation, and filter only the well-predicted metabolites. We can then bicluster this matrix to identify clusters of microbes and metabolites that follow similar interaction behavior. And now we can generate a bipartite graph, as seen on the right. And here the nodes on the left are microbial modules, and the nodes on the right are metabolomic modules. In addition, we identify disease-related modules using Wilcoxon rank sum test, where a module's value is the average feature value of features within that module. We see that the disease-related modules include fatty acids, bile acids, and glycerophosphocholeins, all of which have been shown to have significant impact in IBD. However, we also see interesting non-disease-related modules, such as the green module in the top right, which we believe to represent a large group of amino acids. We also compare the prediction performance of disease status when using the microbial input versus when using the microbial modules, and we observe that there is no loss in predictive performance. This indicates that even though MiMeNet had no information about disease status, it was able to organize microbes into meaningful modules that could still capture disease status. So in conclusion, we have developed a framework called MiMeNet, which uses paired microbiome-metabolome data to learn microbe-metabolite interactions in a data-driven approach. It trains neural networks to identify well-predicted metabolites from microbial data, and uses the trained neural networks to construct a module-based microbe-metabolite interaction network. We believe MiMeNet can facilitate the identification of the metabolic activity of microbes and their association to host disease. Thank you for listening, and if you have any questions, feel free to email me or Professor Yang Dai. I'd like to thank my advisor, Professor Yang Dai, all my lab members, as well as NVIDIA for donating a GPU to be used in this project. Thank you again.